It's my pleasure to present to Shahar Keinan, who is the CEO and co-founder of Polaris Quantum Biotech. Dr. Shahar, with over 50 published peer-reviewed manuscripts, and he is a pioneer in advanced drug discovery. He is in deep knowledge on computational and theoretical chemistry, combined with his extensive business background. He is the foundation for the unique skill set of brings a co-founder and CEO of Polaris Quantum Biotech. Before his success with Polaris QB, Shahar obtained a PhD degree in theoretical chemistry from the Hebrew University of Jerusalem. He received positions at Northwestern and Duke University and served as CSO for Cloud Pharmaceuticals. Thank you, Shahar, for joining today. You can go ahead with your presentation.
Sure, thank you. Let me see that you guys can see my screen. Do you guys see my slideshow?
Yes, we can see it.
Okay, excellent. First, I would like to thank the organizer for inviting me to talk here. This is a really exciting opportunity, and I hope that the participants and the students are gaining a lot of knowledge from these 2 weeks and are going to continue implementing that knowledge in those technologies. So I'm going to tell you a bit about what we do in Polaris and how we accelerate that drug discovery today with QuADD, which is a quantum-aided drug design, and this is a quantum annealing platform. So many things to unpack here: annealing, quantum, drug discovery, QuADD. So let's start. And of course, the first question that we should ask ourselves is, why quantum annealing? And maybe even before that, we should start asking ourselves, why quantum computers? What are quantum computers good for at this stage? So quantum computing solves ultra large scale optimization and simulation problems that are too complex for traditional computers. Let me remind you guys that they only solve some problems. They cannot solve every problem. But when you find a problem that the quantum computer can solve efficiently, you're golden. You can run those calculations much faster. They may crack encryption, which is why everybody's interested in that, speed up decision-making, financial transaction, improve AI machine learning, and some really cool stuff is happening now in quantum AI, in how do we use the nonlinearity of quantum to help us improve machine learning. However, as you guys probably have seen this week, gate quantum computers are still small, and you can call them gate, you can call them fault tolerant. There is really a lot of excitement and a lot of improvement in the last couple of years. But right now, the computers that we have less than 1,000 qubits. And of course, you have to remember there's always the question of not how many qubits, but how many useful qubits there are there that you can use. How many times do you need to run the calculation? How many of those qubits are used for error correction? So these computers are still in development. Interesting work, a lot of proof of concepts, but maybe not something that you can use for production on a day-to-day. And the industry right now assume it's 3 to 5 years from production-ready system. And because of that, the only utility can be found in quantum annealers, not in quantum computers. So even before we go to quantum annealers, this is a really good demonstration of why quantum computers sometimes can solve a problem faster. And if you think about how you solve a maze, okay, if you're using a classical computer, you will need to use that maze, and you walk in the maze until you reach a dead end. You go back to the previous splitting of the roads, and you move forward again until you find a dead end. So you need to scan a lot of the maze, brute force it basically. A quantum computer here behaves more like a wave, not like a particle. It scans the whole maze continuously. Okay, and that's why for some problems, and maze solving is one of the classical problems of where quantum computers are going to be so much better than classical systems. But we are using a quantum annealer, and quantum annealers are different than quantum computers. And the main difference is that while quantum computers can solve multiple kind of problems, they can, of course, solve encryption by factoring large numbers and several other algorithms. Quantum annealers can only solve one kind of problems, and that's optimization problems. They can do it today, but that's the only problem that they can solve. There are several groups that have used quantum annealers to solve large optimization problems today. SavantX, for example, is solving the problem of optimization of the port of Los Angeles. Okay, a port is a big machinery. Ships come in, crane rotate, trucks come in, and they have improved the efficiency of that port dramatically. Quantum South has a way to stack boxes in an airplane. You think about all of those Amazon airplanes or UPS, where you need to put the packages in, you need to balance the weight, you need to make sure that you put as much weight in, and you know that the things are organized in a way that when you reach a certain airport, you just take out some of them, you don't take all of them. So again, it's a large optimization problem. And both of those groups have shown that you can run on a quantum annealer and solve those optimization problems. Now, what we've done in Polaris is we looked at those optimization problems and said, we are interested in drug design, but can we think of drug design as an optimization problem? How do we do that? How do we take a design problem and make it an optimization problem? And at some point, it became clear to us that we can think of drug design as a different problem, which is more of an optimization problem, as part of the backpack. So I'm sure you guys know the backpack optimization problem is you have a backpack, something that you can carry, and you have many items, and you need to choose the combination of the items that will give you the largest benefit, so the best price, but also will be the most lightweighted. Okay, so you're looking for two different properties here. You know, weight, you want to lower weight, and you want to maximize value. So you're doing a multi-object optimization. And I'll show you how we're doing that. But what we are doing is exactly that. We have part of molecules, these are called fragments, and we are finding a way to figure out which are the fragments that together, all of those fragments will create a molecule that will have the right properties. And those properties are, this is part of a multi-object optimization, and I'll show you what the properties are in a short while. So now we're taking all of those things together. We're saying we have fragments that are similar to the items in a backpack problem. And now we have each one of those has properties, and we are interested in the specific properties of each fragment and the total properties of the molecule that will come at the end. And because of that, okay, we can look at a much bigger chemical space or a library of molecules. And then we can think, instead of drug design, drug optimization. What will be the groups that we can add to our molecule that will make it a better drug molecule? This lets us look at very large library of molecule or chemical space. And it's much bigger than any other method that people have been using. And it's called QuADD, Quantum Aided Drug Design. So, however, before we go further, I want you to understand that we are Polaris Quantum Biotech. We're working in a field of computational drug design. In many cases, people ask us not Are there other quantum companies that work on that? But who is your competitor? And our competitor in many cases are other computational chemistry methods. So a bit of a diversion here, thinking about who is using other methods. And to make this a bit lighter, okay, we are saying, okay, let's assume that methods of computation, conventional computings are like dogs, okay? You can think about a supercomputing where many calculations calculate through a brute force solution. If we go back to the example of a maze, then you say, instead of running all of this on a single computer, you run on multiple computers. Every time you reach a diversion in the road, you open another node on the computer and you try that. So this is brute force method. And you can think of that as a sled dogs. All the dogs run in the same direction. There is usually a driver that tells them which direction to go, which is the head node on the cluster. And they're all doing the same work. That's one way. That's supercomputing. On the other hand, you can think of AI machine learning, where there's a lot of work being done on computational drug design. And you train your set, and then you search a data set for a different. Now, the issue with that is that like sniffing dogs, okay? If you teach a dog how to find drugs, they may not know how to find explosives. Okay? So a machine learning algorithm can be good for solving one kind of problem. It may not be good for other problems. So it can find the toxicity of a drug, but it may not tell you if that drug is going to give you the biological activity that you're interested in. Now, of course, since conventional computing are dogs, quantum computing is, of course, cats. Okay, and we can think of two kinds of quantum computers. Again, quantum annealers and general purpose quantum computers, or fault tolerant gate-based. Okay, and the thing about it is, if you think about cats in a room, and there are some places, there's a window with light coming in, the cats will always go to where the sun is. It's the low energy state of the cat, if you would like. So quantum annealing is doing exactly that. You start the system at a certain problem, and the system only relaxes to the ground solution. Okay, you cannot do multi-steps problem. While quantum fault tolerant, general purpose quantum computers, you can do many steps. So you can think of that as training cats for the circus. It's a lot of work, and cats don't like to be trained. But when you have a trained cat, they can do amazing things. The same with quantum computer. First and foremost, it can go down the energy and up the energy. Okay, so you can ask multiple questions. Think of this as the gates that you guys have learned, right? You can do multiple gates one after the other. Quantum annealers, you set up your system and it goes down in energy. General purpose, you set up your system, it can go up, it can go down. It can do multiple gates, multiple kinds of gates as well. Okay, now this is of course a bit tongue-in-cheek, just to remind us that while we're solving problems with quantum computers, we are part of a bigger world of solving problems. And there are other methods out there that we should always think of. So what we do is we ask, okay, what's the ground state of the system? So we start with a protein, which is a big biological machinery. We have a small ligand, which is a small organic molecule, and we throw it into the protein and we see what sticks. Okay, so basically that ligand we just ask, would it fit or wouldn't it fit into that protein pocket? We're not calculating the energy of the system. We're not solving the Schrodinger equation here. What we're doing is we are choosing components and asking, if we combine these components, do we get something that will fit into the protein? Okay, so again, we're only going down in energy. So after I explain to you guys what are quantum computers, what are quantum annealers specifically, let's see how we at Polaris implement this on the D-Wave quantum annealer, which is what we are using. So first thing, and most important here, is that we're not looking for a single property. What we're looking for is a molecule that has many properties. And that's looking at a multi-object optimization. So we're looking, you remember, like the backpack problem? That backpack problem is now we're looking for the combination of parts, of items that will give us the most value, lowest weight. So the same thing you can think of with drugs. We're looking for something that will give us the right biological activity, but be not so toxic, will have the right molecular weight. So we're not going to find one molecule. We're going to find multiple molecules, and they are all on the Pareto front of those properties. So the Pareto front is a set of solutions that all are not, we do not have a single one of them. We have multiple solutions, and the balance between properties is what we're looking for, okay? So for example, this point here, I don't know if you guys see my pointer, but if you do, you can imagine that this point here has a very high objective function 2, very low objective function 1. Here you have in this area something that has a very high objective function 1, very low objective function 2. And this could be interesting, but also all of those things could be interesting because they balance. And this is what we're looking for when we're designing molecules. We're looking for the balance of multiple properties. One of the properties, of course, is how easy it is to synthesize these molecules. If we have the best molecule, but it's going to take us 5 years to synthesize it, it's not a relevant molecule for us. Okay. So we are looking to balance. This is a trade-off among objectives. And in many cases, improving one aspect of the solution will make the other aspects worse. And we're looking to balance those. So this is a Pareto front. And one of the good things about quantum annealers is that you, because you're doing a multi-object optimization, you can choose where you are on the Pareto front, and you can get multiple solutions with different criteria that you're interested in. So we are using the QuBO, which is the quadratic unconstrained binary optimization. That's the algorithm that we use on D-Wave. And basically what we're doing there is we are minimizing an objective based on certain constraint. It's used to solve a constraint satisfaction problem, which means that you are not just you don't have just a single objective, but this objective has to be reached under those constraints. Okay. And as I said, we're looking at the knapsack problem or backpack problem to work with molecular fragments. Here is an example. You have a molecule here, and you can see that you can fragment it. You can break it apart using chemistry rules. This is important so that you can use those chemistry rules. They will let you then recombine the molecules. Okay, if you break it somewhere where chemistry cannot rebuild the molecule, you will get molecules that are not synthesizable. Okay, so these parts of the molecules are called fragments, and they should obey a set of retrosynthesis rules. So they should obey the rules of synthesis. Okay, so here, for the example, for this molecule, you can break this bond easily. There are other bonds that are much more complicated in breaking, so that you will not be able to choose. Now, Each one of those fragments is described by multiple ways. One of them is something called pharmacophore score that tells us how good that molecule or that fragment will fit into a protein pocket. So our objective when we solve a QuADD is to find a combination with the highest combined pharmacophore score. So this would tell us this is the highest value. Okay, so we're looking for a molecule that has the highest value. Each of these fragments contribute to the binding score, to how good this will fit into a protein. However, there are several constraints. For example, when we do a combination, each of those fragments must be one. So we cannot have two fragments sit on top of the other. Okay, that's a constraint that you must include. You have to have certain properties. For example, the total molecular weight, how much all of this weight need to be at a certain value. Okay, the number of groups here that have certain properties. So hydrogen bond donors, hydrogen bond acceptors, groups that are conjugated. All of those things are properties of chemical properties of the fragments that together, when they combine, gives you the right molecule. You don't want too many hydrogen bond donors or acceptors, which means that they will be too soluble. But you don't want none of them because then they will not bind to your protein. So you can, there are rules here. These are the constraints. Okay, and these are the constraints of the fragments, each specific fragments. When we combine them together, we sum over all of those things. Okay, so you see here, if we go back to the QuADD equation, right, and that's the QuADD here is you minimize a certain property, which is the pharmacophore score, but it also has other properties that you need to do that, and it needs to be under the constraints. And the constraints can be either constraints that are inside fragments, so the properties of this fragment specifically, how good does it fit to the pocket, and then you have constraints that are more combination of fragments. Okay, for example, each fragment must be compatible with each connected fragment. So, for example, this bond here has a certain characteristic, chemical characteristic, and you need to make sure that this group and that group can bind. So it's not enough to just make sure that you break the molecule into fragments that are legal. You need to remember what kind of chemistry you have here, so that then those two fragments can connect to one another. Okay. So we're using QuADD. This is the equation. We have objectives, we have constraints. And then we take this and we implement it. And I'm showing you here how it's implemented on the Chimera architectures. There are other ways of doing that. On the Chimera, it's you can see it here. And these are the fragments. And then we say for each of those fragments, how do we connect it to the other fragments? Is there a connection? Maybe there's no connection. Maybe this specific R4 cannot bind to this specific R3. So there's no path here. This R4 can bind to another R3. But if we bind to that R3, are there other connections that we can? Okay. So the fragments are mapped to the quantum architecture. This is important and an important part of making sure that your problem can be implemented or can match the specific architecture of every system. Every quantum system has a different architecture, and you need to think about how you do that. Okay, and then you implement it on the quantum system, and this is how you set up your system, and then you start your system usually at a high energy, and you let the system relax to the ground state. We run this multiple times, of course, not one time, and we got a very large number of solutions, which is what we're looking for here. Okay, we're not looking for a single answer. Again, we are on a Pareto front. We're interested in multiple solutions on that Pareto front. And I'll show you in a minute why. And now when we do that, you can start to think about what is it good for, okay? And I already hinted at that. It's good because it let us look at a very large chemical space, very large library of possible molecules. How big is it? Right now, it's 10 to the power of 30. That's 10 with 30 zeros after it. Okay? Typical search space of classical methods, right? Be those AI or supercomputing, usually look at billions of molecules, 10 to the power of 10. So we can look at a much bigger chemical space, much bigger library of molecules, and that means that we find better molecules. So the molecules that we find are multi-object optimized. So we find molecules that are both have the right biological one, but have the right properties that will make them good drugs. If you think about that, you can take a molecule, a normal size molecule, divide it into 6, 7, maybe 10 fragments. We have a library of fragments that has about 750,000 fragments in it. Now you can do the math yourself, okay? How many molecules, what's the size of our chemicals? It is large. It's 10 to the power of 30. And that's why we get those molecules. And that's why we use quantum computing. Again, using it on a classical computing is going to take much longer. Is 10 to the power of 30 relevant in chemical space, in chemistry? Okay. Maybe I look at 10 to the power of 30 molecules, but all of them are exactly the same and they're not relevant. Okay, so let's look at what is the size of chemical space that we currently people have. Okay, so remind you, the Milky Way has 10 to the power of 11, so billions of stars. The number of approved drugs by the FDA is around 12,000 molecules. So if you think about building an AI or a machine learning model based on approved drugs, it's a very small data set. Okay, but there are other data sets. There are public data sets that collect information from the literature, from other data sets. Okay, there are commercial data sets. These are groups that have built the mechanism of what we're doing on the computer, but they're doing it in real laboratories. So they have a set of fragments and they know how to combine them. Probably the most famous one is enamine 10 to the power of 10. So that's the billion of molecules that a lot of people are searching for. And of course, there's the in-house of Pfizer or GSK or Lilly. A lot of them are private and protected. So we are not sure about how big they really are and how do people use them. But still, you can see that our dataset, our library of molecule is much bigger than anything else here. Okay, and we're using QuADD, 10 to the power of 30 molecules in our chemical space, much bigger than anybody else. So I think I've prepared you enough. Now let's talk about QuADD, quantum-aided drug design. And I'm going to show you how we're building things. And I'm going now to the realm of chemistry and biology. But you, of course, see how this is connected to quantum computing. So we start with a protein, which is a large biological machinery, with a small molecule in the pocket that we are looking to attack. We can do other things around that, but that's where we start for QuADD. And we have our chemical space, our library of fragments that we can use. And then what we do is we build inside QuADD the QuBO. So we're saying, okay, what is our goal here? We want to find ligands or small molecules that will bind to the protein. That would be a binding pocket fit. Maybe we are asking how easy it would be to synthesize those molecules. What's their size and other properties so that they will be bioavailable? And there are additional parameters. So we are building a QuBO that has all the properties that we are interested in for our final drug molecule, right? Not just pocket fit, or not just binding affinity. And the output of that is a library of about 3,000 molecules that are good fit into the pocket, have the properties that we're interested in. I'll show you those in a minute. And they are also in the pocket and in 3D, which is something that people who are looking at those molecules are really happy about. So what we do is I'm going to dive a bit deeper here. This is our pocket. This is the fragment molecule. We can look at it from the molecule that we want to fragment side or from the pocket side. It's the same. We took this molecule, we broke it into groups, and then we search what are the groups that can replace each one of those. And we're using physics here. What's the shape of that pocket? What's the size of it? And what's the electrostatics there? Okay, and that's electrostatic is the pharmacophore that I mentioned before. Now, and we're finding all the groups that can replace this one, all the groups that can replace this one, etc., etc. Now, everything I've described so far is being done on a classical computer. But now I have maybe 20,000 fragments for R1, and another 20,000 fragments for R2, and another 20,000 fragments for R3. And this is where I am reaching a combinatorial complexity. So all the possibilities that I need to look at that are going to be very time-consuming to run on a classical computer. This is where I stop and I say, okay, let's take those lists, let's include them into the QuBO, let's add to the QuBO the constraints that I'm interested in, and the goal, which is binding affinity or fit to the pocket, and build those molecules. Okay, and this is the part that a quantum computer is much more efficient than a classical computer. Running this on a quantum computer is less than half a minute for most of the problems that we're looking at. Running this on a classical computer is a couple of weeks. So you can do it on a classical computer. It's just going to take you a really long time. And you will be surprised, but what's usually happen is that some molecules come out, and somebody would look at them and say, oh, this is great. But let's just see if we can change this, or let's start the whole process again. Instead of looking at our template molecule where we started from, some of the molecules that came out. So this is not done once. And of course, you can fragment your molecule in a different way. You can look at different ways of doing things. So you do this multiple times during a discovery project. As I said, we start on pre-processing on CPU. We use the GCP, but other can. We do the molecular optimization on the quantum computer. Then we do post-processing on the CPU to make sure that some of the things that we get are real. And the output is about the 3,000 molecules that we've shown before. This is QuADD right now is a SaaS, so software as a service. This is how the dashboard looks like. You can see here projects, where they are, who's running what, all of those things. This is how the output looks like. You have the fragments that you started from, all sorts of molecules and their properties, and how they look in the pocket. And just to tell you what the difference is in cost, and this is the cost of running things, not the cost of building the software, not the cost of even pressing the run button. Okay, this is the compute cost. To run 10 targets on the Summit supercomputer is for 1 month is about $2 million, or less than $500,000 for academic pricing. This was a project that was run early Corona times. Some groups use the Summit computer to run 10 targets and find molecules there. It was a very heavy job, as you can imagine, right? To run 10 targets on QuADD, powered by the D-Wave Advantage, cost about $125 and take 5 hours. Okay, the same size of problem, as we've done before. So you can see what's the cost saving and the time saving and all of those things. It's the just think about all the possibilities of things you can do with a quantum annealer when you ask an optimization problem. I'm going to show you two case studies here. The first one is comparing QuADD to AI results. We chose a test system, we ran QuADD, and then we ran an AI method for computational drug design. It's called BIND. We collected molecules, then we calculated molecular properties outside of the system. Both of these systems can calculate some properties, but then at this point we said, okay, we got those molecules, let's do this as if it was a real computational drug design project. And what you do is after you have your results, either from BIND or from QuADD, is you go and you do a high accuracy calculation. And then we said, okay, let's calculate molecular properties, high accuracy calculation. And we selected the top 100 and they are candidates at this point. Not just molecules, they are better than the rest, they are candidates. And we compare the properties of the two sets, of run and of bind. Okay, and this is the binding pocket of thrombin where we started. So I'm going to start with the first thing, and that's comparing binding affinity. How good are QuADD results, they are in blue, to bind result, AI, they are in red. And you can see that the QuADD molecules are way better than the bind result, okay? You can see that they overlap here, but most of bind is around minus 8.2. Most of QuADD are around 911, 929, so minus 929, and so they are much better. They're over 1 kilocal per mole better than the molecules that AI methods get you. And then, of course, it's not enough to have a good binding affinity. We're also interested in other molecular properties. And you can see here again, QuADD gives you much better results than bind, both on how similar they are in their binding pose and how easy they are to synthesize. That's the SS score. So our molecules are much easier to synthesize than AI. So we've done, this is it's just been published this week, those results, but they are a comparison really of the same thing, just how you design a molecule, okay? And comparing AI versus quantum systems. And the next case study I want to show you is you can use QuADD for many things. One of the things is to build and break IP mode. So let's take a look at sorafenib. It's a known drug, a known cancer drug. It took them about 6 years from the time that they decided that this is an interesting biological target that they're going to look at till they started clinical trial. Now, when they started clinical trial, everybody around them said this was a completely new modality in cancer treatment. Everybody said, okay, this is a good drug, new modality. Let's see how we can break the IP of this molecule. This is from Bayer. So everybody, of course, looked at this and say, what can we do? A small change, right, that can give us better molecules. Now, when you write the IP of a new drug, you don't write the patent just for this specific molecule. You look at this molecule and say, okay, what changes, easy changes I can do and would still give me the same properties, right? I can maybe change this chlorine to fluorine. Is it easy enough to do? So you add all of those changes and you look at all possible changes and you write the patent in a way that will protect not just your molecule, but other molecules that are similar to it. Okay, and that's called an IP moat. You build a moat around your molecule so that nobody else can break your moat. Now, of course, chemists are smart people. They looked at sorafenib. They looked at the moat and they started looking around. And of course, they found ways to break that moat. And they wrote patents on those new molecules or publications. And, 25 years later, we came in and we said, this is a good case study for us. We took sorafenib and we said, okay, let's find molecules using QuADD and see if any of those molecules are molecules that other people have found. So what we've done is we looked at sorafenib, and I'm showing you here just one group that we looked at. There are other groups, of course. And we looked at this group and said, aha, this is a change of that group that Bayer didn't think of, but somebody else thought about and wrote a different patent. Okay, so each one of these is a different patent that somebody else wrote and broke their IP, broke their moat. But we, 25 years later, came and showed that we can do the same thing using QuADD. So using QuADD, we could have told Bayer originally, right? We could have told them, this group you need to include this and this and this in your patent. And then these three things wouldn't have happened. But also, if Bayer was now writing the patent, right, they need to figure out what's in it. But also, we could have come to the group that wrote this patent and told them, look at this, look at this, look at this. So you can use QuADD because it looks at such a large chemical space or library of molecules. You can find how to build a better moat or how to break a moat in order to be second to them. So these are two case studies that I've shown. I got instructions to give you guys homework. You can choose if you want or don't want to do them. That's okay, of course. But if you want to learn more about quantum annealing, we've published about 3 years ago a paper that's an example of how to solve a combinatorial problem. The applications are in drug design, but these combinatorial problems specifically is choosing a menu, how you have multiple components of that you can make, and how you make a menu out of those components that would have maybe low calories, less meat, these kind of things. So it's a really neat example. You have a GitHub there as well. Both the paper and the GitHub are available. If you want to learn more about QuADD specifically, there's a paper that we just published, comparison of small molecules generation methods in structure-based drug design. And this is the comparison that I've shown, AI versus quantum computing. Now, if there are chemists here that are interested in working on QuADD, we have a beta testing program. If you are interested, go to demo.quadd.bio to register, and you can run there. Please don't use your Gmail account, use your EDU account or your company account or something like that. Or send me an email, tell me why you're interested in this. I will let you. This is again for it. You do not touch the quantum computer when you're running on QuADD. We are doing it behind the scene for you. So this is really to figure out how QuADD can work for you in a drug design. So I can talk a bit more, but let's stop here, answer a question, and see where you're going from here.
Thank you, Shahar. That was a very nice talk from you. And thank you also for the homework. So we should proceed with the questions, as you mentioned. Heidi is asking, could you share the story behind how Polaris Quantum Biotech started and what motivated its creation?
Sure. So Polaris Quantum Biotech, we started, I even have a slide, I think. Yes, this is the supporting information. So this is Polaris Quantum Biotech from about a year ago. You see here, half the people are chemists, half the people are from different kinds of computer science, right? Front end, back end, algorithms, all of those things. And we started about 6 years ago. So this summer was 6 years. And Bill Shipman this one here. He is Polaris CTO and co-founder with me. And both Bill and myself worked together at a different company called Cloud Pharmaceuticals. And at some point, we realized that the technology there gets stuck. So we can only look at a certain number of molecules and not more than that. And we realized that in order to be better at drug design, we need to look at a much bigger chemical space. And we started looking at what are our possibilities of looking at a bigger chemical space. And that was something that we were extremely lucky, because at that point, quantum computing was only starting, and there were a lot of programs to help people to realize what can and cannot be done on a quantum computer. So when you ask the questions not of drug design, but of drug optimization, when you are thinking about chemical space in a certain way, then using a quantum computer makes a lot of sense. The company was formally founded in February 2020, and we started from there. We have customers that are biotech and pharma, and you guys have seen we're building a SaaS platform. This is the next step for us.
Okay. Brandon wants to know, how do you balance your scientific background with the leadership and business demands of being a CEO?
So it's completely different, but also completely relevant, my scientific background. I did not start, I did not finish my PhD and became a CEO of a company. I was a research scientist, so I learned how to mentor people, how to guide them. And then I was a CSO of a company where I continued to develop my capabilities, and then I became a CEO. When I became CEO, it was, they take you and they throw you into the deep water. There, I treated this as any scientific problem, okay? You read a lot, you listen to a lot of people, you go and talk with your mentors, okay? Different kind of mentors, right? I could not go to my PhD advisor and ask him about, should I take this investment, yes or no, right? So I had to find a new set of mentors, but also I had to learn a lot. That's where my scientific background, which basically taught me how to ask many questions, how to think about problems and solve problems based on knowledge that I have. And you make mistakes. You make many mistakes. Yep, that's how you learn. You make the mistakes, you accept the fact that you made mistakes, and you move forward.
Sounds like a good advice. Okay. And do you believe quantum computing will soon become a standard tool in pharmaceutical research, or is it still early stage?
I think it's very early right now. There are some things, and people in the pharma industry are using it to solve optimization problems. So we are using it for drug design. There are other groups that are using it for things like supply chain optimization, right? Supply chain is something very big in pharma, and you need to optimize it. There are people who are using it for workforce optimization. So, you have many sites, you need a nurse here and a nurse there. Your nurses have different constraints on which day they can work, all of those things. So you use this for better workforce optimization, for example. So there are many things people are using today, and people are really working hard on making sure that they can use it when fault-tolerant quantum computers are coming in, to use it for computational drug design, okay? So solving the Schrodinger equation. But that's still 3 to 5 years away. Today, it is a useful tool, a very useful tool, but maybe ask different questions. Yeah.
Okay. Drug discovery directly impacts human health. How does Polaris QB ensure ethical and transparent use of AI and quantum algorithm? Asks Diana.
Yes, and this is something that we always should remember, right? As a scientist, as computer scientists, as drug designers, the work that we do may look like it's very far away from the people who are going to use it at later stages, but it's not. What we do today influence what will happen later on. So asking questions like, what are the problems that we can solve today is important. It's something, we can take what we're doing and use it for solving other problems that are maybe not so beneficial. One of the reasons that we chose to do drug design is that we believe that this will lower the cost of creating new drugs and bringing them to the market, and that will benefit people. You can use QuADD, even small teams of scientists can use QuADD to develop and to design new molecules, which means that you can treat diseases that maybe before were not commercially viable, right? Rare diseases of areas of the world that maybe people before didn't thought there's enough money in there, right? One of the projects that we've done is Ebola. Thinking about Ebola, which is a disease that doesn't hurt a lot of the more, rich nation, but it is still a problem in the world, and we need to solve it. So, of course, you have to think about these things and be very conscious about what you're doing.
Very good. Okay. In your opinion, what's the most exciting breakthrough on the horizon for quantum application in life sciences?
So I really think that when we have a bit of a bigger quantum fault-tolerant computer, we can solve the Schrodinger equation, which is a holy grail. I don't know how useful it's going to be, but I think being able to solve Schrodinger equation on a system that will be lifelike. So that's our goal here, is to start a quantum simulation that will go to the ground state and give us the real solution of the Schrodinger equation. Everybody nowadays is using a lot of assumptions when they try to solve the Schrodinger equation. It would be really exciting to see the real solution there. I think that's where, again, a holy grail would be really interesting to see when it's done.
Okay. What makes quantum annealing particularly powerful for solving chemical and molecular optimization problems?
I think it's the size of the system that you can look at. Okay. So AI methods or other methods can look at billions of molecules, and that looks like a large enough system, but we know it's not large enough. We know the number of possible molecules is 10 to the power of 60. So we want to be, we are now 10 to the power of 30. It's not half, right? 10 to the power of 30 is 30 orders of magnitude smaller than the full chemical space. And we are really interested in growing the size of the chemical space and being able to look at more and more molecules. And quantum annealing can give us that. The possibility of solving optimization problems here is unique.
Great. For students new to the topic, how does quantum annealing differ from other quantum computing models like gate-based quantum computer?
Yeah, so I think maybe they missed some part of the talk, but quantum annealing is really looking at one kind of problem, which is optimization, and it is only going downhill. So you start your system at a certain energy, so a certain temperature, and you slowly pull it down. It can only go from where the system started to the ground state. Annealing is, quantum annealing is one say, but annealing is a phenomena that is relevant in many other fields, okay? It means that you hit something up and then you slowly cool it down. The same way we make swords, iron swords, right? You take your iron, you heat it up, and you cool it down when you hit it under constraint. Okay, so and then it goes to the ground state, to the most stable form of iron, right? Still, but so that's what you do here. You start from high energy and you slowly cool your system to the ground state. Because of that, you cannot do gates. You cannot say, okay, I'm asking question, and then I'm asking another question, and then I'm asking another question. You can only ask one question, and that question is the optimization of your system. For annealing, right? Yeah.
Yes. Um, uh, how do you see the relationship evolving between quantum computing companies and traditional pharmaceutical giants?
So I think that's a really, good question, and I'm predicting the future is always difficult. I think there was a time where a lot of pharma companies were interested in quantum computing. I think some of them right now are not so interested in that because it takes so long to those systems to come in. I think and there's a lot of AI, and AI is very useful tool, really useful tool in many other properties and methods as well. So I think right now there are some pharma companies that are more interested in quantum, there are less. A lot more of the smaller companies, biotechs, high techs, these kind of groups are working on solutions that will be relevant for pharma. And I think sometimes the best way is not through the front door, right? Is you basically say, okay, let's take a bit of a, side route, work with biotechs, work with other high tech companies, and then get into pharma from there when you have a right solution to a problem they have.
Thank you. Next question. What are some of the biggest misconceptions people have about quantum computing's role in medicine?
That it will solve all of our problems tomorrow. A, it's not going to be tomorrow. It's going to take a couple of years. And B, it's still very early to know what's good and what's not good. There are some things that quantum computers are very good for. If you think about the process of developing a drug, there are some things that quantum computers can be useful at lowering the cost, making better decisions. But in order to make better drugs, we need better biology to understand, side effects, toxicity, all of those things. This is not something that a quantum computer can help us. A quantum computer can help us choose a better patient cohort for a clinical trial. So we will need less people. The trial will be faster. Okay, but in order to really make better drugs, we need better biology. So quantum computing is a useful tool, but is very limited in what it can do.
Okay, and last question. If you could send one message to young researchers in Latin America who want to shape the future of quantum biotechnology, what would it be?
Because what does it has to do with anything that we're talking about today, right? No, but I think the main thing, and this is me speaking as a scientist, as a researcher, is to find something that interests you, but also something that is useful. Okay, you can plan maybe 1 year ahead, 2 years ahead, especially at a field like quantum computing, where the technology is moving so fast. I cannot tell you today which of the possibilities of quantum computing, of technologies, is going to be useful 3 years down the line. Keep your mind open. Look at many solutions. If you're interested in learning something, make sure that that something is relevant for other things as well, right? So if you know how to write a quantum algorithm, you can implement it on many other systems, okay? So what I would say is try to be flexible, try to work on things that you care about, that you understand that that the market cares about, okay? And, but really don't lock yourself into a very narrow range, but make sure that whatever you do has implication in other things, okay? And it could be that you learn how to write algorithms. That's a amazing tool. And you will need, the fact that you use it on a certain computer doesn't mean that you don't know how to write for other things as well. So be flexible.
Yeah. Okay, so thanks so much for that advice, and as
Thank
well
you
for
very
the
much,
other
guys.
ones. Yeah, and also thank you for your talk, for sharing, for staying with us, and I'll see you later.
Thank you very much, guys. Have a great the rest of your day.